Our immune system is a powerful defense force. It protects us from a constant barrage of threats. These threats include bacteria, viruses, and even our own cells that turn cancerous. One of the immune system's most effective strategies is clonal expansion. This process allows our bodies to rapidly produce an army of immune cells to fight off specific invaders. Clonal expansion focuses on a specific type of white blood cell called a lymphocyte. Lymphocytes come in two main varieties, T cells and B cells. Each lymphocyte has a unique receptor on its surface. This receptor can recognize and bind to a specific molecule on a foreign invader, called an antigen. When a lymphocyte encounters its matching antigen, it gets activated. This activation triggers the lymphocyte to divide rapidly. This process creates numerous clones of itself. These clones are identical and carry the same antigen-specific receptor. This army of clones can then effectively target and eliminate the invading pathogen or cancerous cell. Clonal expansion is a fundamental process in adaptive immunity. This is the part of our immune system that learns and remembers past encounters with pathogens. Let's imagine you get a cut on your hand while gardening. This cut introduces bacteria into your body. Your immune system kicks into gear. Specialized cells called antigen-presenting cells engulf the bacteria. They then display fragments of the bacteria's proteins, acting like wanted posters for your immune system. T cells with receptors that match these bacterial fragments bind to the antigen-presenting cells. This binding triggers clonal expansion. The activated T cells multiply, creating an army of clones. These clones then travel to the site of infection. They release chemicals that kill the bacteria and help heal the wound. Clonal expansion is also harnessed in cancer immunotherapy. One example is CAR T-cell therapy. In this treatment, doctors collect a patient's T cells. They then genetically engineer them to recognize a specific protein on the surface of cancer cells. These modified T cells are then multiplied in the lab through clonal expansion. Finally, they are infused back into the patient. These supercharged T cells can then seek out and destroy cancer cells throughout the body. This targeted approach is revolutionizing cancer treatment. It offers hope for patients with previously untreatable forms of cancer. While clonal expansion is essential for a healthy immune system, it can also have detrimental effects. In some cases, the process can go awry, leading to autoimmune diseases or even cancer. Autoimmune diseases occur when the immune system mistakenly attacks the body's own tissues. This attack happens because self-reactive lymphocytes, which should be eliminated during development, escape and are activated. These activated lymphocytes undergo clonal expansion, generating an army of cells that attack healthy tissues. Examples of autoimmune diseases include rheumatoid arthritis, type 1 diabetes, and multiple sclerosis. Clonal expansion is also a hallmark of cancer. Cancer arises when a single cell acquires mutations that allow it to divide uncontrollably. These mutations can occur in genes that regulate cell growth and division. As the cancerous cell divides, it accumulates more mutations, and some of these mutations may give it a survival advantage. This process leads to the clonal expansion of cancerous cells, eventually forming a tumor. Understanding how clonal expansion contributes to disease is crucial for developing new treatments. Researchers are working on ways to selectively suppress the expansion of harmful lymphocytes in autoimmune diseases and to target and eliminate cancerous clones more effectively. Clonal expansion is a fundamental process that underpins the immune system's ability to protect us from disease. By understanding how this process works, researchers are developing new and innovative ways to harness its power to fight infection, cancer, and autoimmune diseases. For example, new vaccines are being designed to stimulate a more robust and long-lasting immune response by promoting the clonal expansion of specific memory lymphocytes. These vaccines could provide long-term protection against a wide range of infectious diseases. In the field of cancer immunotherapy, researchers are developing strategies to enhance the effectiveness of CAR T-cell therapy and other immunotherapies by optimizing the clonal expansion of tumor-specific lymphocytes. These advancements hold great promise for improving cancer treatment outcomes. The future of medicine lies in our ability to understand and manipulate the immune system with increasing precision. By unraveling the complexities of clonal expansion, we can develop new treatments and therapies that will improve human health and well-being for generations to come.